The overall goal of this protocol is to both detect and isolate apoptotic bodies to high purity.
Apoptotic bodies are the type of extracellular vesicles generated after the induction of apoptosis. They are the largest type of extracellular vesicle family, typically ranging from 1 to 5 micron in diameter. It is now becoming apparent that apoptotic bodies can aid cellular processes such as apoptotic cell clearance and intercellular communication. Therefore, the development of an accurate methodology to detect, quantify, and isolate apoptotic bodies is critical for the characterization of their functions and mechanisms. In this series of experiments, we're going to show you how to detect and quantify and isolate apoptotic bodies from apoptotic samples.
Part A. The induction of apoptosis. To induce apoptosis in cell lines such as THP1 monocytes, first start by collecting the cells. Count cells and collect approximately 10 million cells or as required. We recommend 10 million for Western blot analysis of apoptotic bodies. Aliquot 2 million cells per well of a 6-well plate. Remove the lid from the 6-well plate and UV cells using the UV Stratalinker 1800. Irradiate cells at 150 millijoules per centimeter squared. This irradiation should take approximately 30 to 60 seconds. After the irradiation is complete, incubate cells for 2 to 8 hours depending on the cell line. At this point, cells should exhibit apoptotic morphology such as apoptotic blebbing and apoptotic body formation. Collect apoptotic cells with a P1000 pipette.